Microarrays. So you have already heard a little bit about microarrays in the previous PowerPoint lectures. And what they're referring to is a specific type called DNA microarray, which is commonly referred to as just microarrays in general. But you'll see as we go through these slides, there are a lot more types. So what is a microarray? The first microarray technology, or the gene chip, was invented first in the 1980s. And since then, many different technologies have been invented, and they're utilized for specific biological samples. The gene chips are also known as DNA microarrays or DNA chips now, and this is a snapshot of expression levels of genes. You can look a common number is about 400,000 genes. The microarray is a solid surface at which DNA of interest can bind to, so it's a physical chip, and it allows for the analysis of tens of thousands of DNA sequences simultaneously. And the main purpose of a microarray is to analyze genetic mutations. So genes and genetic mutations, just a little bit of an overview. As you know, genes are instructions which tell your body how to make the protein it needs to survive and grow. And genetic mutations are the basis of our individuality. So mutations in our genes are what gives us the different variations in like hair color and height and stuff like that. And sometimes you have a lot of genes that go into one thing, like height. And all of that is just due to multiple mutations or just one genetic mutation. So why would we want to study these? So mutations can lead to missing or malformed proteins as well, not just to good things, but can also cause bad things that lead to disease. By identifying the mutation, scientists hope to better understand how the human body works. It also helps them understand what is happening when it doesn't work properly. So some genetic disorders like cystic fibrosis, sickle cell anemia, and Tay-Sachs disease, and color blindness, all these disorders are caused by one single mutation of a gene. And other diseases include HIV and cancer, which are multiple gene mutations. So microarrays are really awesome because they can help researchers find these mutations and better study them to enhance the way we treat all these diseases and enhance the medicines we need to treat them. There are 12 types of microarrays. You have DNA microarrays, which measure expression levels of thousands of genes at once. This helps scientists who study diseases and develop medicines, like I've already touched on. chips, which are for viewing microRNA populations and interactions between DNA and protein. You have protein microarrays, which determine protein function in a very large scale, and you can study these protein in parallel. So you can compare them and see, if there's a disease in this protein, then how does that compare to normal protein, and then how do you can detect it better in other people, and then you can treat it better. You have peptide microarrays, which is analysis of protein-protein interactions. Tissue microarrays, which is the analysis of multiple tissues for real-time properties at the time of extraction. So if you are going through, let's say hypoxia, or you're going through something like a specific disease at a certain point in time, you can take a tissue, say you have a flare-up from your diabetes. You can take a tissue from some place in your body at that point in time, and it'll show you a snapshot of the expression levels of different cells and different peptides and things like that. And you have cellular microarrays, which this resembles an immunoassay in that it analyzes properties of living cells during immunoresponses. There are chemical compound microarrays, which analyze proteins that bind to specific chemicals. This is really used for drug screening and drug discovery. You have antibody microarrays, which they're used for detecting antigens. Carbohydrate, which this analyzes proteins that bind to certain carbohydrates, and this is mostly used for allergen recognitions in the human body. Phenotype is the measure of phenotypic reactions of cells in response to a changing environment. This is mainly used in drug development. Reverse phase protein, this is mostly used in clinical trials. This detects altered proteins as they relate to diseases. And then you have the iris microarray, which tracks differentiation of immune cells during an immune response. So you have all these cells that go every which way to form other cell lines that will fight with an immune response in different ways. So the iris just detects the differentiation of those cells. So what are they used for? They're used to study gene expression in large scale, determine protein function, analysis of cellular response to changing environments and different stimuli, study states of tissues at a given time. DNA microarrays are used in clinical diagnostic tests for some diseases. Um, and then they also help determine which drugs might be best prescribed for individuals, because individuals react to different types of medicines, or they react to different types of foods. Say you're diabetic, you have a reaction to one type of food and not another, and you have another diabetic that's complete opposite. They're also used, so when they were first introduced, microarrays were used mainly as a research tool. Not many clinical studies were done. Scientists continue today to conduct large-scale population studies, for example, to determine how often individuals with mutation develop breast cancer. It's most commonly used in cancer research and in the pharmacological treatment of other diseases, so like in medicinal research. It enables researchers to investigate and analyze the expression of thousands of genes in a single reaction and address various issues all at once. They can also be used to study the extent to which certain genes are turned on or off in cells and tissues. So here's an overview. Let's see if this plays.
DNA microarray technology permits an investigator to rapidly determine which genes are expressed by a cell or tissue. To understand how a DNA microarray works, let's consider an experiment that compares the pattern of gene expression in tumor tissue compared to normal tissue. When a gene is expressed, it is transcribed into mRNA. The mRNAs from the two tissues are isolated and converted into complementary strands of DNA, called cDNAs, by the enzyme reverse transcriptase. To distinguish between the two pools of cDNAs, the molecules are fluorescently labeled, red for the tumor-derived pool, and green for the cDNAs derived from the normal tissue. After the RNA is removed, DNA microarrays are used to compare the two cDNA samples. DNA microarrays can contain 60,000 or more different DNA sequences attached in microscopic spots to a glass slide. The different DNA sequences are oligonucleotides of about 20 bases in length. The oligonucleotides represent tiny but unique regions of genes in the genome. The cDNA samples are mixed together and added to the microarray. cDNAs that are complementary to oligonucleotides on the microarray will bind or hybridize with the DNA and thereby stick to that location on the slide. Unbound cDNAs are washed away. A scanner detects patterns of hybridization by sensing the fluorescent signals. In this example, a red spot indicates that expression of the gene is higher in the tumor tissue compared to normal tissue. In contrast, a green spot indicates that expression of the gene is higher in normal tissue than in the tumor tissue. A yellow spot indicates that the gene is expressed equally in both tissues. If the gene is not expressed in either tissue, the spot will not fluoresce. Because each area of the microarray contains a known DNA sequence corresponding to a known gene, the identities of the hybridizing cDNAs can be determined. Using these data, investigators can establish which genes are expressed differently in the cancerous tissue, and thus may be able to design better treatment strategies. For breast cancer, scientists have identified 70 genes whose expression differs dramatically between tumors from patients whose cancers have recurred and tumors from patients whose cancers did not recur. From this information, gene expression signatures have been identified that are useful in clinical decision-making. Patients with a good prognosis can avoid unnecessary chemotherapy, whereas those with a poor prognosis can receive more aggressive treatment.
So this is a really good overview of DNA microarrays, which are the most common type as it relates to breast cancer. So why would we use microarrays over other methods? It's really easy to use. There are so many different platforms that you can use. They're lower cost. So like DNA microarrays, since they've been around for a long time, they're pretty low cost and they're high throughput. So you get a lot of results all at once, and it's pretty cheap to do it. Implementation of statistical methods for detailed analysis. There are a lot of different ways that you can interpret results and then apply them statistically. And then you have a global view of gene expression encompassing an entire genome all at once, super quickly. But you do have some drawbacks. The microarrays only present a snapshot of the transcriptome, which is continuously changing and responding to cellular needs, to different cellular needs and cellular signals. And then it's pretty complex. It makes it difficult to obtain meaningful data with real significance biologically without clearly defined goals or any clearly defined targets. And a lot of this technology is new. So while the well-used microarray technologies like microarrays are pretty cheap, the newer ones can be really costly, like those obscure sounding ones that I showed you there at the end. Okay, and then there is a website I want you to see. Okay, so if you go to the slide and click on this website here, this will show you a little bit of how to analyze DNA microarrays. So it'll take you here and you want to launch the interactive. Okay, and it'll take you through how to analyze. Here's an overview. And it just keeps going like that. And so you get a better understanding of how to analyze the data because it can be very confusing. I don't know if you noticed, but some of the images that in my PowerPoint they look kind of wild. So if you're so confused about what that might mean, this is a really good website to help you understand those things.